surface water, but due to different activities done by human being, this water is contaminated, and nowadays this is a grave problem. In the Chikbalapur district also it's the same. Chikbalapur Taluk happens to fall under drought affected area in Karnataka, and the rainfall is scanty. Water problem happens to major crises not only because for the less rainfall and anthropogenic activities by people, but also due to the improper management of existing water resources. Hence, Chikbalapur Taluk mainly depends on groundwater for its various needs. The present study aims to assessing the quality of groundwater in the study area and identifying the area which are polluted so that appropriate mitigative measures can be initiated. The study area I had already told, Chikbalapur is the eastern gateway of Karnataka. It formed by a bifurcating old Kolar district into Chikbalapur and Kolar district. Chikbalapur district possesses an area 4,246.86 square kilometer, and this is given as latitude and latitude and longitude, north and east, and it's containing the 6 taluks, mainly 6 taluks in the Chikbalapur district, that are Bagepalli, Chikbalapur, Chintamani, Gauribidnur, Gudibande and Shidlagatta. I will show in the next figure. And 26 hoblis, 151 gram panchayats and 1,321 villages. This is the map showing different taluks of Chikbalapur district, that is Gudivande, Gauribidnur, Bagepalli, Chikbalapur, Chintamani. And this is the location map of study area. And this is the some geographical features of study area. That is the rainfall is annual rainfall is 740.56 mm. Taluks receive 66 rainy days in a year. This is the groundwater sample. Primarily, I collected in the year of 2018 pre-monsoon and post-monsoon, post-monsoon groundwater samples in the only Chikbalapur Taluk. That are 12 villages in the Chikbalapur Taluk. That the data collected the above villages, that is Arur, Virenhalli, Ajvara, etc. This is the 12 villages I have collected and considered carried out groundwater analysis for assessment using water quality index. This is for the 2018 pre-monsoon and post-monsoon. These are the 12 locations. And this is the 46 locations, whereas from the Department of Mines and Geology, DC office Chikbalapur, the data of groundwater quality has been collected during only pre-monsoon season of 2012 to 2017. This is called as secondary data in the present study. Figure 4 shows the location map of the groundwater sample points in Chikbalapur district for 46 stations. Out of this, in that 7 stations are included by the whichever 12 stations I collected in the year 2018.
Shriram, you are listening to me?
Ma'am.
1 minute. Sorry to hold you in the between. See, we have 15 minutes for each presentation.
Okay, ma'am. Okay, ma'am. I will complete it.
Kindly do it by that.
Okay, ma'am. No problem. So this is the given data. These are the sample points. These are the 46 villages, and this is the geography of the area. It has 3 important rivers: Palar, South Pennar, and North Pennar. This is the hydrogeomorphological map of study area. The methodology, coming to the methodology for water quality analysis, I have used that normal our environmental engineering laboratory manual for the physicochemical parameters like pH, electrical conductivity, total solids, and total hardness, total dissolved solids, calcium, magnesium, this, and laboratory procedure I used. And these are the laboratory performing test. Coming to the water quality index, there are 2 methods I told in the introduction only: water quality index by Tiwari's method and Bhargava's method. So in the Tiwari's method, what happens? In the Tiwari's method, he will give the all constitution as all constituents were present in the water sample. He will scale in the only one scale. All parameters will be the zeros, and pH will be given as a 7. But in the Bhargava's method, he given as some rating. This is the water quality index status and possible usage of Tiwari's method. If this range came for the water quality, that will be the drinking purpose or irrigation purpose or industrial purpose. He will tell excellent, good, fair, and poor. But in the Bhargava's method, he given the rating to the each and every parameter 1, 2, 3, 4, 5. The parameter depends on the study area and depends on the area. He will rate the parameters. Example: magnesium, sodium, potassium given as less quality. Magnesium we are given as 1, and nitrate and fluoride given as maximum 5 weighting. This is the procedure, and this is the status of Bhargava's method. So results and discussion coming to the this is the total, whichever are the 12 stations I calculated for the year 2018. All the parameters. This is by Bhargava's post-monsoon result. So it is observed that the physico-chemical analysis tabulated in the 5 and 6 pre-monsoon and post-monsoon 2018. In that, the table 6 is observed that except the station 6, that is mainly in the Chikbalapur city, all other over stations, fluoride content in the groundwater sample is greater than or equal to desirable limit. Hence, from the table, few water quality parameters are below the permissible limit for the particular station, while few others are.
Share the table.
Ma'am, here first table I shown na.
Okay.
This
Yeah. Now
one.
you explain. Huh.
Table 6 is showing the fluoride content is there that is greater than the permissible limit. Station 8 is a Chikbalapur district. The fluoride content is 2.36. It's greater than the permissible limit, and the same thing for the station 11 also, it's a 2.34. Okay, ma'am. May I can continue?
Yeah, continue.
Okay. So this is the post-monsoon 2018 results. So this is the procedure whichever I explained in the methodology that gives the, see, in this table, ideal value for the only pH is 7, all other values taken as a 0. This is the standard and very accurate method for the Tiwari's method to calculate WQI. But in the Bhargava's method, he rated as WI is on its own basis. That depends on the study area or the area how is affected by the other elements and all. Like if I calculated magnesium as a 1, but another person will come, same study area will take, he will see the magnesium will be, he will give 2 or 3. It will affect on the calculations. So it's a very random method for the calculating WQI. Hence, on this basis, I prefer that the Tiwari's method is a very good results comparing both methods in the study area. Tiwari's method gives better result than the Bhargava's method. This is the WQI by Tiwari's method and the Bhargava's method. See, in this, almost all the parameters will be the good and excellent only. Only one is a Chikbalapur town, that mainly city 8 is a 61.47. It's a fair. This is calculation of WQI by Tiwari's method post monsoon result. This post monsoon result. And these are the status, how the percentage of samples will come under the good, fair, and excellent. See, in the Bhargava's method, almost 50-50, same sample I calculated for both methods, but almost 75% samples came good only in the Tiwari's method, but in the Bhargava's method, both are 50-50. This is the graph. This is the Bhargava's method. That earlier graph is a Tiwari's method, which show the Chikbalapur town is a greater result. This is the Bhargava's and pre-monsoon and post-monsoon comparison. So, whichever I earlier told na, that Tiwari's method is better from the given results. And this is the dental fluorosis notified in the study area, because in the study area, northern part of the Karnataka is a very most and mostly affected by the fluoride content. It's a very high in the greater than the permissible limit, that gives the yellow teeth, nose and bow legs, which are in the figure. It's showing stages of fluorosis and all. These are the agriculture irrigation water quality status during the pre-monsoon 2018. Comparing to the domestic purpose and irrigation purpose, there is no problem for the water quality analysis. Only for the domestic purpose, the fluoride content is more in the study area. This is the table for that 46 stations I calculated WQI. That will show the graph. This is the variation of WQI within the period from the period 2012 to 17. Each taluk I divided that the graph, how the variation of parameters. This is the overall results whichever I got from the WQI in the year 2012, 13, 14, 15, 16 and 17. In this, almost all the samples came by Tiwari's method. After that, the 12 parameter analysis, I calculated Tiwari's by using Tiwari's method only. I calculated the WQI for all 46 stations. So almost the good quality water came for the drinking and irrigation purpose and industrial. But some are the parameters like Gudibande and Chikbalapur, Chintamani Taluk. There will be the most parameters. Fluoride content is more in the study area that I gave in the This is, ma'am, conclusion. Due to the presence of igneous rock like granite, pegmatite, fluoride concentration is higher in the study area. Fluoride content is high due to less dilution of rainfall and geological features of ground. Hence, the defluoridation plant can be proposed for treatment of water, and continuous monitoring of water quality checking can be done for the done to overcome the fluoride problem. To overcome the fluoride detection, the government has taken initiative to installation of defluoridation plant the district to supply good quality of water, and presently the water is supplying from Chitravati barrage to Bagepalli Taluk, Gudibande Taluk and part of Chintamani Taluk for domestic purpose. As the quality of groundwater for calculated WQI during pre-monsoon and post-monsoon, pre-monsoon of 2012 to 17, for irrigation point of view, Tiwari's method is suitable for the irrigation purpose. And these are some recommendations. So for the primary data, groundwater quality analysis was carried out, and it is found Tiwari's method shows better result than the Bhargava's method, because in the Tiwari's method, the weights all parameter are ideal values are considered as 0, and for pH is considered as 7. Also, these weights considered for ideal values for the same for all study area. Whereas Bhargava's method, rating should be given for the each parameter that varies from area to area. This is the subjective in nature. Thereby, Tiwari's method is considered better in concluding WQI than Bhargava's method. Hence, further analysis in the study area has been done based on the Tiwari's method. So, these are the some recommendations, ma'am. It is necessary to adopt the land terracing in the turn help in the increasing infiltration during rainy seasons. Necessary to desil the tanks present in the study area to tanks recharging sites for groundwater. These have been neglected totally by not desilting from several years. Due to this, some tanks in the study area turned into flat land surface, and lots of vegetation has also grown. These are the some references.
Okay, Sriram.
Thank you, ma'am.
I have a few questions.
Tell me.
See, throughout your presentation, you use Bhargava's method and Tiwari's method.
Correct, ma'am.
It is your work in the methodology. What I feel is okay.
Huh, huh.
So since it is your work, only in the methodology you could have just mentioned Tiwari's method,
Huh.
and you could have carried out the work. So
Correct, ma'am. Huh.
in all the slides, you were just comparing Tiwari's method and Bhargava's method.
Hmm.
So you could have kept the topic as comparative study for
Okay.
to study
WQI,
the
water
water
quality index.
quality water quality index by
Hmm.
Tiwari method and Bhargava's method. You could have
Okay,
kept
okay,
the
okay,
topic
okay, okay.
like this.
Title name. You
Title,
are
because
telling. Huh.
see, since it is your work, so
Huh.
I
Okay.
feel that you could have just mentioned in the methodology that you are carrying
Okay.
out only the
Huh. Huh.
Tiwari's method. Okay,
Huh.
one more thing. See,
Okay,
water
ma'am.
quality index. See, it is one of the very the best parameter to
Okay, ma'am.
determine the suitability of water for
Correct.
domestic and irrigation
Irrigation
purpose.
purpose. Okay.
Okay, see,
Huh.
you have taken only physico-chemical characteristics you have studied.
Huh, ma'am.
See, even water is a carrier of all type of microorganisms.
Okay,
You
okay,
could
okay.
have carried out the coliform test also.
Oh,
As a civil
correct.
engineer, as an environmental engineer, this coliform bacteria, it plays a very vital role in water.
Okay.
So just by chemical
I
analysis,
will, I will take out that, ma'am. I will note that point and I
yeah.
will.
See, now you are telling that WQI, yeah, for some
Mm-hmm.
water it is
Physico-chemical
considered,
parameters.
yeah, for physico, just by considering physico-chemical parameter, you
Okay.
cannot say it is suitable for consumption and also for the irrigation. For the
Okay, okay.
irrigation, it is not a problem since in
Huh.
the objective you have mentioned domestic purposes.
Purpose. Correct, ma'am.
So
Correct.
even you should include the coliform
Coliform
bacteria
bacteria.
also.
Okay. I will
Okay.
note that point, ma'am.
So, and then see, in Tiwari's method, you're telling you have given the weightage. The Tiwari has given the weightage,
Huh,
maximum
ma'am.
weightage for pH. And
Huh.
for all other
Zeros.
parameters, the weightage is zero.
Zero.
On what basis Tiwari sir has taken that? Did you do any literature survey on that?
Ma'am, pH is a neutral. pH is neutral. They taken as 7 because the range
Why
of pH
pH?
is
How
6.
can you say pH is neutral?
Because then only the water quality index we can calculate, ma'am. If in
No,
the
no, no.
Bhargava's method, he given as a rating to each and every parameter. I told
Uh,
now magnesium has a 1 and that total hardness he considered as low. The
yeah.
magnesium he given as 5 and total hardness given as only 1. If I
Hmm.
will do the same study for the Chikbalapur district, I will analyze or I will rate the each and every parameter my own analysis.
Okay,
If
see,
another person also same do
so
na,
no,
hence for
see,
that
when
Tiwari given as all zeros to each and every parameter. This is only
wait,
the main source for WQI, ma'am.
yeah, that's what Bhargav.
Tiwari method,
See, yeah, you see, Sri Ram. So
ma'am.
Tiwari sir has taken 7 pH 7. There
Hmm.
is some criteria for that to take because like WQI. Have you heard of
Huh.
AQI? AQI, Air Quality Index. In a
Okay, okay.
similar
AQ.
method,
Huh.
air
Huh.
quality index can also be calculated by taking the criteria air pollutants. Okay.
Hmm. Okay.
So what will happen when you're determining WQI and AQI is you
Hmm.
have to give the weightage for each parameter.
Correct, ma'am.
That weightage, how to give, see, you have taken, this is wrong actually. What I feel, you have taken pH as 7, high weightage you have taken. You should have taken, you should have given highest rating for fluorides because in your area, in your study area, Fluoride's concentration is high, not the pH.
After result and analysis only we got na ma'am, fluoride is high or not.
Yeah, listen.
Because in the Chikbalapur study area, no more papers we got.
Yeah, listen,
Correct.
Sriram. Listen,
Okay.
Sriram. See, WQI is calculated mainly on the rating basis, or the
Correct, ma'am.
we call rating and weightage. Either of the terms you can use. Okay.
Correct,
So how we give the rating, how we give the weightage is which is highly pollutant. For that we give the high weightage. And you said Bhargava's method, there they have given for magnesium 1.
Okay,
why they have given magnesium as 1 is it is the least pollutant. Magnesium is nowhere harmful to human beings or for the consumption. So before, like before taking the Tiwari's or Bhargava's method, what I felt is you could have still collected some more literature on some more papers on the Tiwari and Bhargava's method. Okay. So just by randomly, by taking so it has pH as 7, I don't think it's the right option.
No, ma'am. In that I
Just
studied
go
that
through,
book Chow.
Sriram, just discuss with your guide.
Okay, ma'am. Okay.
Just discuss with your guide. Okay. So I feel taking pH as giving a high weightage for peers, for pH might not be the right option. Okay.
But in the all literature they took pH only 7, ma'am. In all papers, how many papers I studied for the literature? Around 80 to 90 papers I studied for the literature survey. Everywhere pH took as 7 only in Tiwari's method. His procedure is same only, ma'am. If we want to give the rating for each other parameters like fluoride, magnesium, then we have to study only Bhargava's method. On the Bhargava's method only we have to tell W2.
Okay, you could have looked for some other international journals. They have collected other methods. Okay. So I'm talking about the international papers. They have used many methods. So and
other methods are there, man. Index method is there. Smith method is there.
See, otherwise, what you could have done is you could have taken the combination of two methods. See, it is your research paper. Okay. Otherwise, you could have done the combination of both Bhargava's and Tiwari's method. So what I felt is okay. You should have included the coliform bacteria, and you could have tried the combination using combination of both Tiwari and Bhargav. Okay. Asha, do you have any questions?
1 minute, Sriram.
Okay, please.
Sriram, I have a question for you.
Okay, thank you.
Is it MTech work or PhD work? Hello, your work is it MTech work or PhD work?
MTech work, not
MTech, no? You completed your MTech or what you are doing? Work, sir. Yeah, presently you are doing it.
Completed. Sir.
You are completed. You are ready.
No. I finished my MTech. I'm working.
See, here there are several problems. Number one, what you have done the physico-chemical water quality analysis, where you have not done the ionic balance. First failure is that one only. First do the ionic balance, because what is the total hardness? It is a combination of calcium hardness plus magnesium hardness. Calcium you have given 70 or magnesium you have given 30. Totally it should be 100, maybe 105 or 110, something like that. But whereas total hardness you have given is 200. Somehow abnormally you have written the results. That's what I can tell. It is not the analyzed one. Please try to verify your results, because authenticity is required, where the water quality analysis plays a major role. At the same time, you also told that no water quality reports are available in Chikbalapur districts. I am sorry to tell that. Several, I mean, you can refer any books. Either it may be from the agriculture department or from the engineering department. So many studies have carried out specifically on water quality issues on Kolara district and Chikbalapur districts. You should do the thorough literature survey. At the same time, I think Shivana is a geologist, no? Dr. Shivana,
sir.
Yeah, you can have a discussion. You sit with sir. Sit with Shivana sir and discuss, because he can guide you properly, number one. At the same time, better to use the Piper trilinear methods. Whatever the water quality analysis, Piper trilinear method, you can easily.
1 second.
Yeah.
Sir, 1 second. 1 second you can tell, sir. I will note.
Yeah. Piper, Piper, trilinear methods. Trilinear.
Okay.
Trilinear methods. Generally, all geologists will tell whenever you are going to talk about the water quality, we have to follow that guidelines laid by them. Okay. Anyway, of course, you have made an attempt. Good luck. Fine.
Thank you, sir. Thank you, sir. Thanks.
Asha, ಹೇಗಿದ್ದೀರಾ ಮೇಡಮ್ ಚೆನ್ನಾಗಿದ್ದೀರಾ? ಸಾರಿ ಐ ಟುಕ್ ಯುವರ್ ಟೈಮ್. ನೀವು ಮಾತಾಡಿ ಪ್ಲೀಸ್ ಕಂಟಿನ್ಯೂ. ನಿಮ್ದು ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಂಡಿದ್ದೀರಾ? ಮ್ಯೂಟ್ ಮಾಡಿದ್ದೀರಾ ನೋಡಿ ಮೇಡಮ್. ಮ್ಯೂಟ್.
Thank you, sir. First of all, thank you for making me the session chair.
ನೀವು ಬಂದಿರೋದು ನನಗೆ ಖುಷಿ ಆಯ್ತು ಅಷ್ಟೇ. ನೀವು ನೋಡಿದಂಗೆ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿ just we requested you. Continue.
Yeah, Sriram, it is very nice work. And how did you estimate the fluoride contamination in the water, Sriram? What method did you use for that? Sriram, you can hear me? Sriram? What is the method you used for evaluating fluoride in the groundwater? AS method, huh? Your voice is breaking. Soumya, you are there? Voice break ಆಗ್ತಿದೆ ಅನ್ಸುತ್ತೆ ಸೌಮ್ಯ.
ಹೌದು.
Absorption spectrometer.
Yeah, okay, good. Okay.
Yes, ma'am.
Okay, nice. Good, Sriram. Okay, many questions have been asked. You answered good. From my side, no questions. Good work. Kindly make the suggestions given by the other professors and include it in your thesis. It will be okay. With this, we can move on to the next presentation. Soumya, can we move on?
Yeah. We shall move to the next
presentation. Thank you.
Good effort. Good work. Okay.
Thank you.
So now I invite.
Thank you, ma'am.
You're welcome. Now I invite Hansa K to give presentation on isolation and screening of antimicrobial peptides. Hansa?
Hello.
Can you share?
Hansa, you're there?
Yes, ma'am, I'm there, ma'am.
Yeah. Load your presentation, Hansa.
Yes, ma'am, I'm just loading. Ma'am, is it visible?
Yeah, it's
start
the presentation.
So, good afternoon, everyone. I'm Hansa from BMS College. I'm doing my MTech. So my topic is isolation and screening of antimicrobial peptides. So first, what are antimicrobial peptides? Antimicrobial peptides, these as the name suggests, antimicrobial. So these inhibit other microorganisms. These are also known as host defense peptides, and these are present in all forms of life. Even yes, ma'am.
Sorry for the interruption. Can you go to the next slide? Still, it is in title.
Oh, no, ma'am. I've gone to next slide.
No. Here it is not changed, Hansa.
It's not changed.
Yeah. Okay. Now it is. Now it is.
Now continue.
Okay. Yeah. So so the antimicrobial peptides, these are also known as host defense peptides. These are small cationic peptides. These can be isolated from variety of sources like your microorganisms, plants, even animals. Now, why do we want to work on this antimicrobial peptides? Now, these antimicrobial peptides, they are antibiotic, and they are of broad spectrum, meaning they can kill your bacteria, virus, fungi, even cancerous cells. Now, why do we want to work on this? One problem is multi-drug resistant microorganisms. One problem is this. Second problem is your biofilm forming bacteria. Now, what are biofilms? Your bacteria, they form layers. When they form layers, they secrete a very slimy layer called polysaccharide. So now the antibiotics, they are not able to break through these polysaccharide layer. Now how do these antimicrobial peptides work? Now what they do is they specifically target the cell membrane. Now this is how they work. This is the mechanism of action. So first, what there are three models. One is toroidal pore model, barrel stave model, and carpet model. So toroidal pore model, what happens is, in all the three models, first the antimicrobial peptides, they attach themselves to the surface of the membrane. Then in toroidal pore, what they do is they make the membrane curve inwards. That causes a gap in the membrane and causing disrupting.
Can you move the slide? Can you go to next slide?
Ma'am, on the next slide, it's not visible.
No.
No, you're not moving the slides,
See
Hansa.
only the second slide. Yeah.
Is it now? Is it?
Yeah, yeah.
And now also is it visible?
Mechanism of action. Yeah, you start with this.
Okay, okay. So this is the mechanism of action. So this is the toroidal pore model. So in all of the three models, the antimicrobial peptides, they attach to the surface. So now in toroidal pore model, it causes it moves the membrane inwards, causing a gap in it. So this disrupts the membrane, causing cell death. In barrel stave model, it causes a pore. It attaches to the membrane and causes a pore in between, again causing cell disruption and cell death. In carpet model, they attach themselves on the membrane and they till a point where they reach the threshold, that is sufficient amount of antimicrobial peptides when they attach to or bind to the membrane, then they cause a pore as shown in the figure, disrupting the cell membrane, causing cell death. Now these antimicrobial peptides, ma'am, is the next slide visible?
Yeah, it is.
So these antimicrobial peptides have been extracted from various sources. For example, this peptide, bacteriocin, it has been extracted from lactic acid bacteria. Your thionins have been extracted from wheat endosperm. Defensins from tissue, seeds of potato, heaven-like peptides from latex of rubber tree, nautin-like peptides from fungi, insects, and pycidin from Olignathus fasciatus. So now these are the currently available antibiotics like penicillin. This is used for respiratory tract infections. Then you have tetracycline. This is used for acne or respiratory or genital infections. Then you have aminoglycoside. This is used as ear or eye drops. Now, these are the microorganisms that are resistant to the commercially available antibiotics. Like you have Neisseria gonorrhoeae, then you have E. coli, then you have Acetobacter baumannii, Staphylococcus aureus, Pseudomonas. So these are some of the microorganisms that have developed resistance to these antibiotics. Now, how do we proceed is, first what do we do is we take different samples, like mixed different samples, like even food waste, your cow dung, many things have been mixed. Now, then you take zero dilution. We haven't diluted the sample. You're taking it directly and you're putting it on a medium. The medium used here is nutrient agar medium. So you allow the bacteria to grow. So you incubate it at 37 degrees for 24 to 48 hours. Now, when you remove after 28 to 48 hours, what you see is you check for a clear zone, as in this picture. Ma'am, is the picture visible?
Yeah.
Yeah. So here their markings is 1, 2, 3, and 4. So in the first plate, you can see in between there's a microorganism growing, and around there's a clear zone. So the bacteria that is growing in between, it is not allowing the bacteria around it to grow. So it has inhibited the other bacteria. Similarly, for the number 2, this bacteria is not allowing this bacteria to grow around it. Similarly, for 3, this bacteria isn't allowing this bacteria to grow, and this in between bacteria is not allowing the other bacteria to grow. So what we have done is, the bacteria which is not allowing the other bacteria to grow, where the numbers have been put, we have isolated them. We are subculturing them. So we cultured those 4, and we have maintained them in something known as nutrient agar slants, so that we can use whenever we want. Now what we do is we screen these 4 samples, isolates that we have got, against disease-causing microorganisms. Now the disease-causing microorganisms that we have used here is E. coli, Pseudomonas aeruginosa, and Staphylococcus aureus. So now what we are doing is, first so here the growth medium that is used is known as Mueller-Hinton agar. So we on each plate, so suppose you're taking first plate, so you spread E. coli on it, and then you're making 5 wells. One is for your blank, that is distilled water, and the 4 isolates that we have got from isolation. Those 4 isolates you're pipetting into the wells. Now then you incubate them for 24 to 48 hours, and then when you remove, after 24 to 48 hours, you check for the zone of inhibition. if there's a clear zone. Similarly, this was done for Pseudomonas and Staphylococcus. So what was seen is E. coli, E. coli was inhibited by the second isolate, and the second isolate having the maximum zone with 15 mm, and sample 1 showed the least inhibition with 11 mm. Similarly, Pseudomonas, it showed second highest inhibition among the all three, with sample 3 with a zone of inhibition of 18 mm. Staphylococcus aureus, sample 3 did not show any inhibition against Staphylococcus aureus. And against sample 4, it showed the maximum inhibition of 19 mm. Now we want to know if this inhibition that is being caused by the bacteria, if it is caused by a peptide or no. So for that, what we do, we use ammonium sulfate precipitation. Now what does ammonium sulfate precipitation do is, when you add the salt to the solution, ammonium sulfate salt to the solution, now what protein forms, protein is bound with the water molecules. Now when you add ammonium sulfate, it competes with the protein for binding with water molecules. This reduces the solubility of protein, causing it to precipitate. So now how this is done is, so you take your sample, you prepare a broth of the sample, that 4 isolate, you prepare a broth, and then you centrifuge it for 10 minutes at 7,000 RPM. Then whatever supernatant that you get, that you collect, and then you make up the volume till 100 ml. Now the amount of salt that is required for that volume, we calculated using precipitation chart. This is the precipitation chart, for example. Now if you're taking the first, so what we do is we take for 20, 40, 60 and 80 concentration. Now at the start, there is no ammonium sulfate added, the concentration of ammonium sulfate is 0. Now we are doing it for 20%. So now for 20%, here you can see the top column, for 20%, the weight is shown as 114. So you're adding that much, so that this table is for 1000 ml. Now what we have made the volume till 100 ml. So you have for 1000 ml. So you can calculate how much of salt is required for 100 ml. Similarly, you calculate it for the subsequent steps. Now, after calculating how much of the ammonium sulfate is required, you add it to the supernatant, and then you mix it on a magnetic stirrer with a stir bar placed in it. Now, and this is to be done at 4 degrees because proteins are very sensitive to temperature, so it has to be done at 4 degrees. So now, after that, after it is completely mixed, you take the sample and you centrifuge it at 10,000 RPM for 10 minutes. Now when you centrifuge, you'll get supernatant and pellet. Now you take the pellet, dissolve it in phosphate buffer, and keep it at 0 degrees so that you don't, to prevent the denaturation of protein. So that is your protein. Now the supernatant that you have got after the centrifugation, you take that, and to that now you add salt, ammonium sulfate salt again. So that would be a 40%. So now for 40%, how you do is, now already you have saturated the solution with 20% salt. So now you look for 20% of the salt, and this is the salt concentration. So you take 20% of the salt, and you're doing this for 40 saturation. So 123 grams of the salt is to be added for 1000 ml. So you can calculate for 100 ml. Similarly, it is done for 60% saturation and 80% saturation. Now what we do is, after you get, now for each of the concentration, you'll get a pellet, that is your protein. So that you have stored it at 4 degrees. Now what you do, now we have to check if this protein is causing inhibition. So again, you'll do the well diffusion method. You'll take your test organisms, E. coli, Staphylococcus, and Pseudomonas. Then you pipette your protein sample that you've got into the wells. Now the other E. coli and Pseudomonas did not show any inhibition, but for Staphylococcus aureus, the inhibition was seen for these percentages: 40, 60 for sample 1, 2, and 4. So this showed that yes, so the inhibition that was seen against Staphylococcus, that was caused by a protein. So what we can say by this is, the proteins that the proteins that these microorganisms produce, they can be used as an alternative to the conventional antibiotics. So there you also tackle the problem of multi-drug resistance. You can also eradicate your biofilms, because these E. coli and Staphylococcus and Pseudomonas, these are biofilm producing bacteria. So you can inhibit them, and you're also tackling the problem of multi-drug resistance. And since these are selectively, specific to the microbial membrane, so there is very less chances of them developing resistance to these peptides because they disrupt the membrane itself. So they can be used as a potential alternative to your conventional antibiotics. Yeah, thank you. Hello?
Yeah, thanks, Hansa. Now, where are you carrying out all these experiments?
Sorry, ma'am?
Where are you carrying out these experiments? Can I know that?
Ma'am, this is...
Where are you carrying out these experiments?
Ma'am, this is done, ma'am.
You finished this. Where did you do this?
In which laboratory? Your college have this laboratory, this microbiology laboratory?
Hello?
Yeah,
ma'am.
Hamsa, we can hear.
Hello.
From where
Hello,
did you
ma'am? Yeah.
Where did you carried out this work? In which laboratory?
Yes, ma'am.
Hamsa?
This is my... yes, hello, ma'am.
Yeah, tell me.
Hello, ma'am, I'm audible.
Yeah, you are.
Yes, Hamsa, we can hear your voice.
Yeah, this is my fourth year project, B bachelor's project. Hello.
Okay. So,
yeah, ma'am, this is my bachelor's project. Hello.
Okay.
Where did you carry out this experiment, Hansa?
Hello.
Hello.
Ma'am, this is my bachelor's project, ma'am.
Correct. Where did you carry out this?
Hello. Yeah, this is
which laboratory?
MBIT. This is my bachelor's project. This is my fourth year project. So I've carried out Sir MBIT. Hello.
Yeah, I know Sir MBIT. MBIT has this.
Yes, ma'am. There. Yes, ma'am. It has.
Do they have from Hansa? Do they have this sophisticated lab to carry out this experiment?
Yes, ma'am. They have. I've done my B in biotechnology, so I've done this there as my B project.
Okay. Hansa, this colony counter, you did it by colony counter. How did you isolate it? What was the methodology? Which equipment? Is it colony counter to find out the number of isolated antibiotics?
No. I didn't use colony counter. What we did was we took the sample. Suppose you have mixed the sample. So we spread it on a plate.
Yeah.
So when you spread it on a plate, so there will be many microorganisms. So in that step itself, we are seeing if there is any bacteria present that is inhibiting another bacteria that I showed in the image.
Yeah, how did you see that? Through microscope?
No, ma'am, you can see it by naked eye. You'll get a clear zone around this thing.
Yeah, only by color identification. Color identification?
No, ma'am, you'll get a zone. Ma'am, I'll show the people. One sec, I'll show the picture, ma'am.
Show me that. That I didn't understand when I was seeing yours.
Here.
Yeah, this slide.
Ma'am, in the image, you can see numbers, ma'am. I've numbered 1, 2.
The colony.
Yeah, so that one in between you can in between you can see a very prominent spot. So that is one bacteria, and around it you can see a zone. So that bacteria is not allowing the bacteria around it to grow.
Okay.
Yeah.
So this is your antimicrobial peptide. How will you distinguish it? How will you come to know this is not any other microorganism and it is only that specific microorganism? How will you come to know?
I didn't get it, ma'am. Specific?
Yeah.
Yeah.
See, you have marked 1, 2, no, on each colony.
Yeah.
So, how do you figure out this 1, 2 are only AMP?
Ma'am, because the zone is only around that bacteria. Other places you can see it is fully like grown out. Only there it is like there is no growth around it. So that is the bacteria that is causing the inhibition.
Okay, answer. In the next slide, go to slide number 10.
1 second, ma'am. Yeah. Yes, ma'am.
Yeah, you said this will be maintained for some time. What is the life of this cultured bacteria? What cultured?
Ma'am, this can be maintained for a long time. Just it should be made sure that it isn't contaminated. So you cover it with a cotton plug. If it gets contaminated, then it has to be thrown away.
Oh, okay. So how long what is the life of this? No idea. You can maintain it for how long, however long time.
Ma'am, I used it for 3 months. 3-4 months it was there without contamination. We had it for 3-4 months.
So okay, so this is your B project, not your MTech project.
No, ma'am.
Okay. Okay, Hamza. Thanks. That's all from my side.
Okay.
Any questions from students? Okay. Hamza, good
Yes, ma'am.
presentation. So we wish you all the
Thank
best.
you, ma'am.
Okay.
Thank you, ma'am. Thank you.
So now, Asha, shall we move to next presenter?
Next we will go for next presenter.
Okay. So now I invite Hemalata ji to give presentation on sewage water treatment using vetiver grass. Hemalata? Hemalata, can you hear us?
Yes, ma'am. It's not audible to my voice.
Can you share these slides and start your presentation?
Yes, ma'am.
Hemlata, your voice is breaking. Your voice is not audible, Hemlata.
Good afternoon. Working in a farm industry, assistant professor. So first of all, I would like to thank the management of Dayananda Sagar College of Engineering giving this great opportunity to us, and thank chief patrons, honored chiefs, and organizing committee members.
Hemlata, your voice.
Sewage water treatment using vetiver grass.
Hemlata, your voice is not audible, ma. I think there are some background noise. Hemalata, we could hear the background noise. Asha?
Yeah.
Asha, ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಇಲ್ಲ, background noise ಇದೆ ತುಂಬಾ. Hemlata, you are there? Can you hear us, Hemlata? Hemlata? Soumya, I think there is some disturbance in the audio.
Yeah. Hemalata? No audio.
No audio.
Hemalata?
கேக்கல, வாய்ஸ் கேக்கலாம்னு சொல்லிட்டு இருக்கீங்க.
Can we send her a message in the chat box?
I think she has not started the presentation.
Yeah, she has not started also. I think there is some problem in her audio.
Hemalata, please respond. We cannot hear your voice.
Soumya, can you ask the coordinators to call her?
Asha?
Yeah, Soumya.
Nitin is contacting Hemalata.
Okay. We will wait.
Just a moment.
Yeah, we will wait.
Hello, ma'am.
Yeah.
I'm devising in my mobile, ma'am. Please, sorry.
Yeah, Hemlata, you're audible now.
Yeah, ma'am. In mobile only, I'm connecting. Actually, previous in laptop only. So I think it may some issues there. Just please wait. I'm opening my slides.
Yeah.
Okay. I think she has a microphone problem.
Yeah.
Yes, afternoon.
Yeah.
I'm Hemrata, working in SRM University as assistant professor in SRM Ramapuram campus. First of all, I would like to thank the management of Dayananda Sagar College of Engineering giving this great opportunity to us, and thank chief patrons, honorary chairs, and organizing committee members. So my title is Sewage Water Treatment Using Vegetable Grass. Firstly, what are all the overview? The project is introduction project. It is the sewage water treatment. It is having the more methodologies there. The what is the penetrations process and what is the secondary permanent treatment process is there. It is also have the it is the major source of the recent years of the water pollution has become the most serious and environmental problems. And having the one more important the topic is that is the vetiver grass. Vetiver grass, what is the major source here? Vetiver grass means it is the ability of what vetiver grass to act as a primary purifier. Ma'am,
can you change the slide? It is in the first slide only.
actually I am changing only. Just wait. Yes. So everyone see the introduction of this sewage water treatment. It is the World Health Organization reports that more than 80% of the diseases affecting the humankinds are waterborne diseases. Why? Because why the major source of this sewage water treatment? What is the major source we are doing the sewage water process means it is the direct health hazard to those people who come in physical contact with such waters. So we have to purify that waters. Then so most now in this generation more are the harmful according to this not purifying water. So groundwater can be contaminated due to the disposal of this inadequately treated wastewater, and it having some reasons there that is identical to make properly treat and dispose of wastewater always. And here the major the title of this major agenda is the wetland technique. It is a very new technique here which helps to treat the water economical at a core level. So next slide, what are all the advantages? What are the benefits of this sewage treatment? It is the cost efficient in terms of construction, operations, and maintenance. And the next one, effectively treats wastewater from human waste, agriculture. So not only the human waste, it is also useful for the agricultural, and not only agriculture, stormwater, some metals, pollutants, mining, and industries. Low energy consumption required for operations, prepare water for reuse. What is that reuse? The water is the bad water. In that water we have to purify it, we have to filter it, then it contaminates the all that ingredients in that water. Ingredients all will dead decomposes in that purification of the reusing water. And assisting maintaining contributes to environmental protection. These are all the advantages of the sewage water treatment. Next one, and it having some disadvantages also there. In any project, in anything, the advantages either that is for benefits is there and disadvantages is there. We have to identify what are all the positive and negative, and then only we have to start that our project or whatever. So here in this title also some disadvantages there, that is the maintenance for cleaning and clogging issues. Breeding mosquitoes, permanent land is requires expert design and supervision technique. So see here, it is a permanent land required. So why as a civil engineer we have to construct a building, we have to check first thing is the site, the permanent land, we have to choose that land. So according to that we have to in this title also we have to check the permanent land is required. That either that is the wetland requisitions or normal land, we have to check the permanent land. After we have to checking, then we have to finalize the that particular land permanent land is requisition. So here in this title some objectives there, the efficiency of the wetland treatment process, natural resources of water treatment, reuse of treated wastewater, flood protection to find out the wastewater properties after treatment process. See here, these are all the 5 objectives. The one is the wetland treatment process. What are all the benefits? What is the positive points in that wetland techniques? We will discuss in the next slides. And water treatment process also. Yeah, the water purifications is having more treatments. We all knows very well that is a primary treatment, secondary treatment, and tertiary treatments. Like that, we have to check the treatment processes, reuse of treated wastewater. So before slides we discussed the reuse of water. So like that, we have to check the bottom of the subsurface of the water surface, reusing, retreated. How we have to check that it is one of the equipment, and it is the objective flood protection. If any flood protections comes with, this is also one of the objective to identifying the what is that problem, and to find out the wastewater properties of the treatment process. So after completed that all three treatment processes are the tertiary, sedimentary, and primary, these three treatment processes completed, while finally we have to check which is the best one, which is the suitability. Next one, literature. So in this title we knows what are all we all knows what are all the literatures, how many literatures, and in that each literatures what is that characteristics. The first literature is the Danya. So in that is the phytoremediation study, the constructed wetland technology. The one that is the literature review, the very most important, the first one is the constructed wetland technique. Yes, right, very good. In that constructed wetland technique, using what is the major purpose here? Vetiver grass only we are using, that is the first, and it is in this vetiver grass total dissolved solids, biological oxygen demand, chemical oxygen demand, nitrates. So chemical, more number of chemicals there. We have to check that vetiver system, that bottom subsurface vetiver system, what are all the chemical names and how to test that. That is a one major description is there in that the literature review. That is the first Danya, Jivan, and Jaya. Next one, multi applications of vetiver grass, that is for Jagannathan Kantaraj Raman. So what he discussed, that is for conservation projects and commercial projects on vetiver cultivation. So same vetiver grass, previous literature review, in that what are all the chemical compositions we have to need, we have to use that there in that previous slide. But in this literature review, is there any conservation projects, any new cultivations is there, like that any byproducts, not only the chemicals, any byproducts also there. Several vetiver conservation projects in future, like people and environmental having making some situations is there, means how to rectify. So that one is the second one, the second literature review of the Kentoraj Raman bioenergy and the energy Jagannathan sir. And the next third literature review, that is the K.M. Bivire and K.N.J. Roy and R.J. Minja. So in
Madam Hemlata.
Yes, tell me, ma'am.
Madam, kindly go to your work now. Literature review is okay. Kindly go to your work.
Okay. Yeah. Okay,
your contribution you
so
kindly.
Is this yeah, okay, tell. Okay, ma'am. So here the methodology. What are all the methodologies we are using in this title? Means here, see, already we discussed the primary treatment process, and so primary treatment process. So in that sewage water treatment, the bottom surface water treatment, how to rectify, what are the treatment processes? The one is the primary treatment. The primary treatment is the process of removal of large and heavy particles such as cans, plastic bottles. These are all the main particles is there in that primary treatment, and it consists of temporary holding of the sewage in a basin, and in primary sedimentation stages, the pre-settling phases. These are all the points under primary treatment. Coming to the secondary treatment, is the substantially degrade the biological content of the sewage and human waste also. So previously that is for cans and wastages. Now second treatment is the substantial degrade, that is a human waste, and the secondary treatment process, it dissolves the suspended biological matters also. And the third one, tertiary treatment, it provides the final treatment stage. So from starting stage to that final treatment stages, all influenced, and that all further improvement quality, that is for sea, river, lake, and wetland, ground, etc. It is the receiving environment discharging, that is the tertiary treatment, and more than one of the tertiary treatment process may be used at any treatment plant. So construction wetland. So what is the major in this title, how to construct this wetland? A constructed wetland is an artificial wetland created for the purpose of treating anthropogenic discharge such as municipal or industrial. It may also be created land reclamation, ecological disturbances. Constructed wetlands are engineered system, natural functions of the vegetation, soil, and organisms. And in these wetlands designed to emulate futures natural wetlands, such as acting meals from the water. And here in this construction wetland, we are discussing.
Hemalata, have you developed any pilot plant set up for this?
Yes, ma'am.
Yeah, yeah,
Yes,
please,
yes,
please
yeah.
go to that and explain.
Yeah, okay, ma'am. Okay, this is the subsurface flow wetland, and so what are all the materials we have to use as sand, soil, and coarse aggregate as collected? Vetiver system. So vetivers also we are collected in the treatment plants. So this is the vetiver process. What are all the materials used in that materials? How to make it that setup? So there are 3 layers setup. The one is the coarse aggregate, and the second one is the sand, and the third one is the vetiver. How to experiment? So experimental setups here, a pilot scale experiment was set up. This test to the effect of the vetiver on the removal of the pollutants sample. So sewage plant is constructed by using 150 liters of PVC drum. The first is the primary treatment, and the second wetland, the third one territory tank. So design step, how to design. The design step is a sewage plant is constructed by using 150 liters PVC drum. In that drum, coarse aggregate up to 20 centimeters height from the bottom of the PVC drum. That is the first layer from bottom. The second one is the second layer with the sand is filled in the drum up to 50 centimeters height from bottom of the PVC drum. Listen, this is the design step. The 150 liters PVC drum from bottom 20 meters height. Second layer is the sand filled, that is up to 50 centimeters from bottom PVC drum itself. Above that, the soil is filled up to 80 centimeters from the bottom of the PVC drum, that is a vetiver grass. So finally, how it look like, we will see here. The first is coarse aggregate, second one is a fine, and the third one is vetiver setup. So everyone see it in this setup. The third diagram, in the third diagram, the bottom from bottom itself only, each and every layer compacted. Finally, vetiver system is here. Test conducted. What are all the tests? That is for pH, BOD, COD, dissolved oxygen, and total suspended tests. So what is that? pH and biological chemical oxygen demand, dissolved oxygen, total suspended solids, and each having how to calculate. And dissolved oxygen having some formula. Based on that formula, and these are after completed that treatment with sewage treatment, we took that values and we tested based on this chemical pH, COD, DO, BOD, total suspended solids. Based on that formula we are using, and test results. See here, initial, the day 2, day 5, and day 8, the pH value is 6.7, and dissolved oxygen having no range, and BOD 260, COD and TSS having this, and that is the starting step. And the next second, day 2 is 6.9, 1.1, 215, 425, and 250 TSS. Like the day 5 values and day 8 values. Here the graph variations, pH value, how that variations, DO, how that variations, BOD, COD, and TSS, each having. So pH, the starting is initial stage 6.9. Already we saw in the table form. Based on the table form, we created these graphs. Initial stage 6.9, the pH is there. Day 2 is increasing, and day 5 it's increasing, and day 8 it increasing up to 7.6. The range of this pH value is 6.5 to 7.8. So the next DO is like this. Initial is nothing, and the day 2 is from 1.2 DO values. COD graph variation, initial stage, day 2, and day 5, and day 8. BOD values, and last TSS values. Based on that graph representations, we done that all 5 methods. Based on that methods, these graphs will represent. And finally, my conclusion is the roots of the graphs were found out to the main aspects in the purification of process. The main thing, purification of water, and the second one, in this tested, it is having gap of 3 days, gap of from initial day to gap of 3 days. It compared to the other days, just according to the changing the time, and it result shows that the water contamination is less, and the water is used for various household purposes. And thus we conclude that our project is success. We have the success value, that is CPCD. It is the Central Pollution of the Control Board of India. This is only my project. Thank you.
Yeah, Madam Hemlata.
Yes,
this vetiver gas, what is grass? What is the life of the grass? How many times you can use the sewage water in that?
Actually, one time only we use that. The main ability of the vetiver grass to act the primary purifier of the wastewater. Only one time only we are used, and it having some low cost sanitation also.
How much grass can filter? How many liters of sewage water?
About 4 meters, ma'am.
4 meter by 4 meter, you are telling. Hello?
Ma'am?
Yeah, what is the how much surface area of the grass can filter how much sewage water?
Hemlata, did you sow the seeds of vetiver grass, or directly did you use the plant with the vetiver plant, vetiver grass?
Actually, we collect. No, ma'am. We went in Kodalur, ma'am. Collected that vetiver grass.
Okay, you collected the grass and you used. Okay.
We went in.
You placed soil and then you treated.
In Kodalur place. Yes, ma'am. In Kodalur place, we went and we collected that vetiver grass. After the vetiver grass, first we have to fill that layers, 2 layers. After then third layer, this one we have this vetiver grass we kept.
Madam, what is the contribution of vetiver
grass? Yes, ma'am.
See, your coarse aggregator and sand will be used as a filtering media, something like rapid sand filter or slow sand filters. What will this grass remove?
Slow sand filter.
Now, what is the contribution from the grass?
Actually, this
removing?
Yes, ma'am. Actually, the main contribution of this vetiver grass is the high tolerance of the harsh conditions, ma'am. And any harmful, if any harmful soil is there, also it is very tendency is there in this vetiver grass. And this vetiver can be used in the variety of the different ways also. Either that is for treat substandard water and effluents also. It is used for the major source in this vetiver grass.
Madam Hemlata, my question is what is that pollutant that grass is going to inhibit? What pollutant it will stop? Now sand and coarse aggregate, we will know suspended solids are getting absorbed in the sand and coarse aggregate. Correct, no?
Yes,
likewise, what is that grass doing? What is that grass resisting?
What is the grass?
What is the contribution of grass?
Sorry, ma'am.
And will take the suspended solids. Sand and coarse aggregate will hold the suspended solids and make the will send the clear water to the bottom of the tank. Now, likewise, what is grass doing?
Yes, ma'am. Yes, ma'am. What is the grass doing?
Yeah.
Hemalata, you never you never discussed here vetiver grass.
Yes, ma'am.
The vetiver grass
does
not remove the pollutant. It doesn't act as a treating media. The roots of vetiver grass, it actually treats. You never mentioned throughout your presentation about the roots of vetiver grass. Is it so?
Roots of presentation. Yes, that word is correct only. But
I feel the root is acting like a filtering media.
Yes. Root filtering media. Yes, ma'am.
Yeah, not the grass.
Right? So what is your findings?
Yes, ma'am.
Madam, what is your findings?
So finally the finding is the purification of water only, ma'am. Based on the purification, it having many sources. This is also one of the source that is the purification of water.
See, what you could have done is, Hemalata, madam.
Yes, ma'am. Yes, ma'am.
First, you should have passed the water through the soil media, the soil what you are using for this project, and after that you could have planted the vetiver grass, and then you could have find out the concentration of the water. So then the contribution of the soil and vetiver grass in treating the wastewater could be easily identified, I guess.
Yes, ma'am. Yes,
okay, madam. It is nice work, ma'am. Thank you.
Yeah. Okay, ma'am. Thank you, ma'am.
Okay, madam. So good presentation.
Thanks, madam. Now, Soumya ma'am, with this we are going to conclude this session.
Yeah, we will conclude this session. So, any questions from the students? Okay, as there are no questions, so we will conclude this session. So all the 3 papers were unique and good. So I thank Asha for joining us for this technical session. Suhas sir, it's over to you.
So on the behalf of organizing department, I thank Dr. Asha ma'am and Professor Soumya ma'am for the valuable feedback and for the sharing this technical session 1. Thank you, ma'am.
Yeah, thank you, Suhas. Thank you, Soumya.
Thank you, Suhas. Thank you, Aisha.
Yeah, we will log out with this. Thank you.
Okay.
Thank you, ma'am.
So now we have a lunch break and the session will resume at 2 p.m. The session, technical session 2 is resumed at 2 p.m. So participant can rejoin with the same link. Thank you all.
Thank you, sir.